The purpose of the presentation today is to present to you what kind of technical services we may be able to offer, but at the same time, we want to get some feedback. What kind, many of you may be thinking about developing a new project, and what kind of a new technique you may be needing. And then you can look at what we do, then you can say, can you develop some assay like this? And I will not say we'll have all the assay, but at least we can try. So this is a listing of the facility we have. So I wouldn't spend too much time reading about, but you can just look at it. Inevitably, this is more focused on the HIV/AIDS testing because that's what we do. But as you know, many of the methods can be easily adapted to the different field. So we can discuss about that. So, for example, we have a reference laboratory with the Roche AmpliTac for quantification. Nowadays, HIV, HCV, HBV, and possibly cytomegalo is coming up, I understand. And this is the automated DNA, RNA purification station, so we can process the sample fairly quickly. We have now 3-flow cytometer, FACS caliber, 4-color, and FACS array 4-color. These are beads capture methods. So you can capture these protein or DNA, RNA on the beads and quantify. So you can look at some protein component, for example, cytokine or chemokine in the plasma during diseases. Or we can look at the messenger RNA expression in the cells. And this is the FACS area 11-color cell sorter. So usually what you people are used to, what I would call static analysis. You look at the marker expressions and how many cells with such markers, etc. But I want to emphasize the flow cytometry is not really limited to the static analysis. For example, I'm sure UCC group also run intracytoplasmic cytokine production. So you simulate the cells and look at how much cytokine, chemokine they do produce, and so you can assess the functional aspect of the cells. And good thing is you can look at for each different subset of the cells. And instead of saying in overall how much cytokine, for example, they are producing, you can say what cells are producing how much. And I will say additionally, there are some dynamic assessment of cell function. For example, there are many other assays, but flow cytometry can also be used for calcium or sodium channel function of the cells. Good thing about this, you can look at the channel function of the different cells. You can tag with a cell marker and analyze the channel function of a certain cell subset. And this is one of the things we have been doing. For example, the PGP and other efflux proteins. So we can look at the efflux function of the, for example, PBMC or whatever the target you want to look at. So this is the sort of display of what we have. We have actually we have now 6 regular cycler, happen to be all ABI 97, 9800, and what is called the new one. We used to use Biolab instrument for the real-time PCR. Now we are totally switched to the light cycle 480, we show you. And again, we started DNA sequencing analysis using the initially visible genetics, Bayer, Siemens now, the long-read tower. But we switch mostly to the ABI capillary sequencer. Now, I'll explain why. And many of you do the DNA amplification sequencing in your own lab. But again, I want to emphasize even the sequencing now has become simply a tool. It's not a purpose. So it is a question of what you are going to do with the sequencing data you obtained, not really to obtain the DNA sequencing. So we can help. We can do the DNA sequencing, but what you do with the sequences is your game, not my game. As I said, 6 ABI cycler, 2 Biolab ABI 7000, and 3 light cycler 480 for the real-time PCR, and 3 Siemens long-read tower, automated DNA sequencer, and ABI 3730 with the 48 capillary. I always say, we don't want to brag, but I end up bragging about our lab has been since 2008 WHO accredited for the what we call the HIV ResNet, which is a global drug resistance mutation surveillance and the monitoring program. And they have a very stringent requirement, and we have to pass the ACTG panel, biannual ACTG panel. Then they impose WHO criteria. And I want to add to the bragging again, we have not only the true gene system, which is the FDA approved kit-based the genotyping system, but we already also have the homebrew system, the WHO accredited. I'm only saying that we're not that bad. So these are the light cycle 480. Actually, this is side, but since we put the sample in this way, so we turn the cycler for our convenience. Some of the issue we have been promoting is and we discussed with some of the UPR group as well. Nowadays, because of the fiscal restrictions, the testing cost become a very crucial issue. For example, the HIV/HCV viral load assay is in the range of probably around $80, $100, $120 per test. And then we do less, but our standardized, validated homebrew protocol probably costs only about $10, $20. We discussed with the CDC, PEPFAR person in the Caribbean.
Dr. Yamamura, what do you mean by homebrew?
Homebrew research protocol. So it's not kit-based. You buy each reagent and you standardize.
So you have to perform that for viral loads? Yeah.
Right. And of course, there are many validation panels nowadays. So you can, when I discussed with the CDC people, their point was as long as it is validated, they don't have any problems. And as a part of bragging, I may add again, for example, since we take the ACTG panel annually, we get the test result of all the participating laboratory, including national, international ACTG lab. And I will not point out the name, but we find very interestingly, some of the big name institutions using FDA approved kit not always pass. And we passed both TruGene as well as the in-house. And in one of the panel, you may be interested to know, because this was maybe about 2 years ago, Siemens lab using, of course, their own TruGene kit failed, and we passed.
What's the minimum number of copies that the homebrew test detect?
With the viral load, it's really the how you run sample. If you have an elaborate sample concentration procedure, it's not really the assay. It's the sample concentration procedure. So because you have to have at least 1 or 2 copy template in the reaction mixture. And we use 20 microliter, 10 microliter template. So how many copy in the... So without concentration, we can go to probably hundreds. The ml, yeah. But, as I said, there are many concentration kits. So if you use it, for example, the reason amplitude goes down to what, 5? Is it? Yeah, 20? Copy per ml is because they use 1 ml of the plasma, and all the elevate goes into one reaction. So we typically use 150 microliter or 200 microliter, and elute in the 50 microliter and use only 10. So we can change the assay protocol to increase the sensitivity. And we actually started this homebrew approaches because it's more crucial issue is as you know, the genotyping I don't know how much UCC currently charge, but we say maybe current market price in the USA is probably between 200 and let's say 400 to 450. And I know the Clinton Foundation testing, sending to South Africa for $600. And with a homebrew using WHO accredited protocol, we have been offering for example, Trinidad and some other Caribbean country at 50. And we're not losing money. And we can, for your research protocols, these are not really Mickey Mouse assays. As I said, it's WHO accredited, BQA, proficiency tested annually. And we can introduce into the research protocol. And for example, in 2009, Pan American Health Organization signed a contract with us to provide the clinical genotyping. They pay $100. So we also have fish microscopes. One applied, one the inverted. We just upgraded the camera, so now we have a much high power digital camera which we can swap back and forth. So for the many DNA or even just a fluorescent, you can get a much better picture with this system. And we also run the GC mass spec for the illicit drug. Well, it doesn't need to be only illicit, but even illicit drug analysis. Well, unfortunately, we have not gone through the accreditation for the drug identification. So, as you know, the drug or substance abuse is one of the factors you have to always consider in the health problem of the patient population in Puerto Rico. So the not only illicit drug, but licit, as well as including alcohol, I suppose. We demonstrated taking the sample from the street, saying that for example, while most of the interview-based analysis showed the large majority of drug users are using speedball, needle analysis actually showed the pure cocaine was the large majority. And last year we published with the Vargas Build groups the abuse of the veterinary sedative among the injecting drug users of Puerto Rico. So, as I said, the sequencing is a sequencing, but the question is what you can do with the sequences. This is not our data, but this is from Tampa Group's publication, looking at the migration of the HIV in the patient over time. So these are necropsy samples, and virus migrate. So as I said before, it doesn't have to be HIV. you can look at other pathogens, but you can look at the movement using the viral sequences. And we use the same technique to look at what particular virus compartment is responsible for heterosexual transmission. And we demonstrated that the different T cell subset of the patient have different HIV strain. And we apply the same to look at how the virus, how similar or different the B creating HIV in the Caribbean country, and look at the gene flow migration among the countries. This may give you the sort of molecular epidemiological analysis of some pathogens from among the individual, from population to population, from one geographical area to the others. As you know, these sequencing has been used to even criminally identify the person who passed the virus to the, particularly knowingly, to the recipient. So this is the kind of conclusion we can come up with the molecular epidemiology analysis using sequencing data. So as I said, it's not limited to HIV, many pathogens, or even the individual. So we want to know, for the kind of project you're thinking about developing, what you may think we may be able to contribute. Not only what we are doing now, but using the same techniques, we can develop a new testings. And we already, I think that this is completed. Yeah, we, for the PRCTRC, we prepare a new web page, and so this is going to be linked with the PSME EDU website. And again, for example, the project in the future, if you are approved center project, how much is the charge going to be? You can click on the list, and we do other immunology reference laboratory function. So not the center project, but if you want to use the reference lab function, what tests are available and what are the charges. And again, we can always discuss about it. So this is a kind of listing we have. And also we have this is not showing too well, but the contact information of the person who would be responsible for the web management and all this. So you can go to the person and ask all the questions. And I don't yet, but we'll have for example, the test ordering slip online, so you can fill out and click, and it will go in. Thank you.